Hello students, welcome back. Today we are going to discuss some other concepts from principles of inheritance and variation. Today's first concept is test cross. Definition: the cross made between F1 hybrids with its double recessive parent to conform homozygous or heterozygous condition of F1 hybrid, or is also defined as a cross between an individual of unknown genotype and recessive parent is called a test cross. So let's see once. So already we discussed in previous part, that is monohybrid cross. In that monohybrid cross, we cross the two parents, that is one parent, that is female parent, with a tall character that we are mentioned as T. This is the female parent we are mentioned, and a male parent, that is small t, and small t, it is a recessive character. So they produce the gamete with T, and male produce a gamete with a small t, and during fertilization these two are fused to form F1 hybrid. F1 hybrid. So now test cross, it is explained that a cross between an individual of unknown genotype. Suppose you don't know this one genotype, that means the genital composition you don't know. Then what we need to know, the unknown genotype is need to cross with a recessive parent. So parents, this is one parent, female parent, and this is the male parent. So among these two parents, we need to take a recessive parent. So that is a recessive parent with the small t, we are considered as the recessive. So the unknown, that means whatever the F1 hybrids are produced, that F1 hybrids we need to cross with a recessive parent. Then we confirm that whether the F1 hybrid is a homozygous or maybe heterozygous. So test cross is defined as a cross between F1 individuals with double recessive parent, that is small t, or whatever the character you are taken in that double recessive parent with F1 hybrid. If you did that cross, then it is called as test cross. So let's see the significance of the test cross. Test cross is used to test whether an individual is homozygous, that means pure, or heterozygous. So the importance of this test cross, why they are doing. So here on paper we are mentioned that dominant is capital T, and recessive is small t, and F1 is capital T, small t. But in field it is different. We are observing only phenotypes. That means when tall and dark plants are crossed, the all F1 individuals are tall. That means we are saying only phenotype, external morphology. If you want to know the genotypes, so when these F1 individuals are crossed with a recessive dwarf parent, then we should know about whether the unknown individual is homozygous or heterozygous. And it is also used as a test for linkage. So that linkage concept we will discuss in this chapter later. There are two possibilities of test cross. One, one pure dominant violet parent plant. It is represented with capital V, or as per our test book, it is mentioned with capital W, capital W. So violet parent plant with capital V, or maybe capital W of unknown genotype plant is crossed with a pure recessive plant, pure recessive white plant represented with a small v, or maybe represent with small w. In such a cross, 100% plants will be violet as in the normal monohybrid cross. So once again, when pure dominant violet plant, violet parent plant is dominant violet of unknown genotype plant is crossed with a pure white recessive plant. Let's see the representation of this. So here, dominant phenotype that we don't know as per question. A pure dominant, that is capital W, or maybe capital V, that we know, pure dominant one, and it is crossed with a recessive white plant, that is homozygous recessive. So that we are saying that it is a small w and small w, and here unknown genotype we are mentioned with capital W, or you may also taken capital V, capital V. Here also a small w, or you may use a small v also. Then these are the gamete two. It is a capital W, capital W. Two gametes are having capital W, and here two gametes are small w, small w. So when this is crossed, all are heterozygous. Capital W, small w, capital W, small w. Here capital W is a dominant gene of the violet color. So that's why in these offsprings all are violets. So the result is all are violet flowers. So unknown flower is homozygous dominant. So when this 100% of plants are appeared the same phenotype, then that indicates that the unknown one is the homozygous. For example, if you are say that, sir, I don't want to that, the unknown one, you are imagine that unknown one is a heterozygous. That is second possibility. The unknown one is heterozygous. That means capital W and small w. Then what will happen? Here it is, one is capital W gamete and another gamete with a small w. Then what happens when the capital W is fuses with these gametes? These two are the heterozygous. In small w, it start with the fuse with that 2R. Here the capital W becomes small w, and here capital W becomes small w. Small w indicates that white color. So in this case, one unknown plant is heterozygous when it is crossed with the recessive white plant. Then you will get a 50% of plants are violet and 50% of plants are white. So that indicates that if 50% are recessive and 50% are dominant, then it is called as the unknown phenotype is heterozygous, that is capital W, small w. If all are 100%, then unknown one is homozygous. That means capital W and capital W. So let's see the second possibility, when hybrid dominant violet. Hybrid dominant. So here the hybrid dominant is nothing but it is heterozygous. Hybrids are having characters of both the parents. So that's why here hybrid dominant, or we can say that it is a heterozygous. So when heterozygous dominant violet or hybrid dominant violet, capital V, small v, or capital W, small w, plant of unknown genotype is crossed with a pure recessive white, that is small v parent plant. In such cross, 50% plants will be violet and 50% of plants will be white. So let's see that. So same. So here the previously here it is unknown one is a dominant, but here the unknown one is the dominant but hybrid. So that means heterozygous. So this is capital W, small w, or capital V, small v. But it is crossed with homozygous. Definition is clear. The cross made between unknown one always with the homozygous, recessive or double recessive. We need to cross unknown one, any unknown individual, either homozygous dominant or heterozygous dominant. Whatever the unknown individual is always cross with recessive parent. Then when you will get 100% of dominant characters, 100% or dominant, dominant, 100% or dominant. That is, here the 100% of dominant character is violet. It is a dominant one. So all the individuals are violet, then it is unknown individual is with a dominant character. That means genotype capital D, capital W, or maybe capital V, capital V. So as per here in this statement, half of the flowers are violet. That means if 50% of the individuals are dominant, that is violet 50%, and 50% of individuals are white, then it is called as this unknown one become heterozygous, that is capital W and small w, or maybe capital V, small v. So I think you may understood properly. So test cross is nothing but cross made between unknown individual with the recessive parent, or directly we can say that the test cross is defined as the cross made between F1 individuals with double recessive parent is called test cross. So that is useful to know whether the unknown individuals are homozygous or heterozygous. That is the significance. The next concept is backcross. Definition of backcross. The cross made between F1 hybrid with any one of the parent is called backcross. So already we discussed the previous concept, test cross. In test cross, the cross made between F1 hybrid with the recessive parent only, with the recessive parent. But in back cross, the cross made between F1 hybrid with any one of the parents, either with the dominant parent or with recessive parent, is called back cross. And this back cross is also having two possibilities, because F1 hybrid cross with the dominant parent, then the available offsprings are different, and F1 hybrid cross with the recessive parent, then the available, that means the offsprings formed during that cross are also different. So there are two possibilities of back cross. One, there is a cross between F1 hybrid, that is capital T small t, capital T small t, and a dominant parent, that is capital T, based on the monohybrid. So in such a cross, plant will be 100% tall. Let's see that. So see, F1 hybrid already we mentioned, that is capital T small t, and you need to cross with a dominant PP for parent. So that dominant parent indicates that capital T, capital T. So here in this mentioning dominant, here the P represent the parent. So parent is dominant, it represents with the capital T, capital T. So we are very well known that hybrid, F1 hybrid is heterozygous, capital T small t. That's why two different gametes are formed. But dominant parent, it is a homozygous, capital T, capital T. So the gametes are having same gene, that is capital T, capital T. So then when the cross this takes place between this, that is F2 generation, you can identify that all plants are tall. In that 50% plants are homozygous tall and 50% are heterozygous tall. But phenotypes, all four plants are 100% tall only. So this is one possibility. That is, when F1 hybrid is crossed with the dominant parent, all individuals are dominant character, that is tall. That is one possibility. The other possibility, that is, there is a cross between F1 hybrid with a recessive parent. So then in such case what happens? So F1 hybrid that is always capital T small t, and a recessive parent, that is a small t small t. So then the resultant is two plants, that is 50% of plants are the tall plants, and remaining 50%, that means two plants are dwarf plants. Dwarf plants. So F1 hybrid when it crosses with the recessive pair. So we need to clear about that difference between test cross as well as back cross. So in test cross, in test cross, in test cross, this test made between unknown into recessive parent, it is already fixed. This is a test cross. Test cross is always unknown one with recessive parent, that is test cross. Whereas in this back cross, in this back cross, the test, it is made with always F1 with individuals with either dominant parent or the F1 hybrids with recessive, then it is called as back cross. But test cross is always unknown one with recessive. That's why due to this test cross, we need to know the significance is to know the homozygous or heterozygosity of unknown one. So we are doing this test cross to know the genotype of unknown one, whether the genotype is homozygous or heterozygous. We can identify by using this test cross. Please beware of it. The next concept, law of dominance. Based on inheritance of one gene concept, the law of dominance can be summarized as given below. Various hereditary characteristics are controlled by factors nowadays called as genes. Factors occur in pairs. The factors contain the information about the form of trait or character. That means factors contain or genes contain information about character. Out of these, only one factor express itself in the F1 generation. The factor expressed in F1 generation is called dominant, and one hidden or suppressed as a recessive. The recessive factors appears again in F2 generation. So this all summarization is completely depends on previous video, that is one gene concept, mono hybrid cross. If you thorough with that monohybrid cross, it is easy to understand. So once again, I am repeating these points again. So various hereditary characteristic. Hereditary, we knew very well that that is transferring the characters from one generation to another generation is controlled by various hereditary characters are controlled by it is factors. So nowadays the term given by Johansson, the factors are called as nowadays genes. Why we are using factors? Because Mendel used that word factor instead of gene. Later that factor named as a gene by Johansson. So the various hereditary characteristics are controlled by factors or genes. So these factors or genes occur in pairs wherever you are writing. Suppose female, the genes we are expressed with XX, or male we are mentioned with XY. It is always with a pair. So factors or genes occur in pairs. Next, the factors contain the information about the form of character. That means trait is nothing but a character. So genes contains the information about the character. That character is transferred to the next generation. Out of these characters or out of these genes, so a pair of gene, only one factor express itself in the F1 generation. So now once recollect your monohybrid cross for this point number 4. So in monohybrid cross we are using two parents, that is one is a dominant parent, that is capital T, and a recessive parent, that is a small t. When these two are cross pollinated, so the gametes of these parents are one is capital T gamete with and another it is a small t with a gamete. And when these are fused together, that means it consists capital T and small t, and this resultant is the F1 generation. So what we said, all F1 individuals are tall. All F1 individuals are tall. Once recollect, what is the genotype of this? Capital T, small t. Generally, small t it is a representation of the recessive. Capital T is a representation of the dominant. So here in F1 generation, both the dominant gene, that is capital T, and recessive gene, that is small t, are present. But the phenotype, the expressed character is tall. That means all plants are appear as a tall plants. That means the recessive character is hidden, suppressed by this dominant. That is explained in this point number 4. Out of these, that means out of these, this pair of factor, only one factor. So out of this pair, that means these two factors, only one factor, that is capital T, in F1 generation expressed the character, that is tallness, but one is hidden. The character which is expressed in F1 generation is called dominant, and which is hidden is called as recessive, and this recessive factor again reappears in F2 generation. So here it is all about the representation of the dominance by using monohybrid cross. So we are all very well known that that is pure tall parent. It is crossed with a pure dwarf parent. So pure tall, it is represented with capital T, and recessive, that is character, represented with small t, and capital T is the dominant. Now they form the gamete with capital T, and recessive plants also produce the gametes with the small t. During fertilization, the F1 individuals are originated, that is capital T, capital T, capital T, small t, that is F1 generation. Now, all the F1 generations all are tall plants. So now what we discussed in this law of dominance. So all the factors, genes are present in pairs, and these factors are responsible for the character. That means we are saying that capital T responsible for the character of tallness, and small t responsible for the character of dwarf. So now here in this gametes, the gamete with capital T, that means this gamete carries the tall character, and this gamete carries the dwarf gamete. So during fertilization, they are restored. The pair condition is restored during fertilization. So then after fertilization, F1 generation is formed with the capital T and small t. In this F1 generation, only one factor, that is gene, is expressed, that is capital T character is expressed. That character is called as dominant, and which is hidden, it's not expressed, that is called as the recessive. So this recessive small t character is expressive during F2 generation. That all we are discussed in previous slide. The law of segregation. Definition of this law of segregation. The law of segregation states that allelic pairs separate or segregate during gamete formation, and the paired condition is restored by the random fusion of gametes during fertilization, is called law of segregation. The law of segregation can also be stated as in other way. The hybrids or heterozygotes of F1 generation have two contrasting factors of a dominant and a recessive type. They do not contaminate or mix with each other. They separate or segregate at the time of gamete formation, so that a gamete carries only one factor, either dominant factor or recessive factor. Children, be careful while reading this statement. Please try to represent the monohybrid cross flow chart, that is capital T, small t, and what are that hybrid or maybe F1 generation. That all you need to represent on the paper. Then you can clearly understood this concept. Until unless without work on this. Yesterday itself I told that genetics is nothing but a puzzles. You need to practice with a pen on paper. Otherwise, it is always puzzle. So please be careful. Try to write on the paper what you understand. So here it is. A female a parent, that is with capital T, small t. Already we are saying that it is also say in other way. In that we are taking that F1 individuals. So because if you consider the parents of this starting, that means before, for the formation of F1, F1 individuals are formed by using the parent factors, that is capital T and small t. These two are homozygous. So that's why gametes, these two factors are separated during gamete formation. Two gametes are formed, each one with this capital T and capital T. So now, whereas to clear understand of this, we need to take this F1 individuals cross with F1, that is the selfing of this F1 individuals we need to take. Then we can understand clearly. So now F1 in F1 plants, that is hybrids, the female having capital T small t, and males are also having the same factors, that is capital T and small t. What we are saying that the always factors are occurs in pairs. So in that pair, one pair having capital T and small t and another also same. So they both are self. So now in this, the factor consists a pair of factors with the different trait or characters. So in this pair of factors, capital T represent a tall character or tall trait, and a small t represent the dwarf trait or character. So both are present and they are not contaminated or mixed. And these factors with the different traits are separated during gamete formation. This law is called law of segregation. Segregation is nothing but separation. So a pair of contrasting characters are present in a parent cell. They are not contaminate and they are segregate, that means separate or isolate individually during gamete formation, that we will call as law of segregation. Mendel's law of segregation is based on the following conclusions. Number one, each trait is produced by two factors. The factors may be similar or may be dissimilar. Here similar means that factor, that genes are similar. That means either capital T or maybe small t. These two factors are similar, or maybe dissimilar. That means capital T, small t, dissimilar. Each true breed form possesses two similar factors. So true breeds, already we said true breed is nothing but homozygous. That means it consists a similar factors, that is either capital T or maybe small t. And each hybrid form has two dissimilar factors. So dissimilar factors we can observe in hybrid, that is capital T and small t. But in true breeds, we can observe the similar factors, that is homozygous. Dissimilar factor is nothing but heterozygous. So homozygous condition we can observe in true breeding, and heterozygous factors we can observe in hybrid or F1 generation. And second conclusion, each gamete possesses only one factor of the pair. A gamete is therefore pure for a trait. So now already we mentioned, suppose if you are taken that hybrid F1, that means dissimilar trait or dissimilar factors you are taken, that is capital T and small t. During gamete formation, it forms the two gametes, that is with capital T and a gamete with small t. And this gamete is pure for a character. It is a pure, that means this gamete having only capital T. That means it is consists only character of tall, and this gamete consists only character of dwarf, small t. So when this gamete with a capital T, that is a pure triad, is crossed with another pure triad of the tall, that is a capital T, then the resultant one is homozygous. Otherwise, if it is crossed with another one, that is another pure gamete of the recessive character, then it gives again heterozygous, dissimilar. So here each gamete possesses only one factor of the pair. In a pair, only one gene is received to the gamete, and it is pure for that character because it consists only one gene. That gene represents its own character. And third conclusion, there is a random fusion of male and female gamete to form a zygote. So fusion leads to the formation of gamete. So random fusion. So then fourth conclusion, the offspring receives only one factor from each parent. So already we know that. So parent plant form the gamete, and in that each gamete, one is from the male parent and another one from the female parent. Only one factor is received, and the factor do not blend in F1 generation. Blend means do not change in F1 generation, but retain their individuality. A recessive factor is not altered by a dominant factor. So here in F1 generation, in previous, that means F1 generation, all the individuals are tall. F1 individuals genotype representation is capital T and small t, and it is even though recessive gene is present, all the plants are tall. Clear? The factor is not blend in F1 generation. So here the factor, that means gene, does not change here. Both are present together, and this recessive gene is not altered, that means contaminated or mixed by this dominant factor. They are present in a cell and they retain their individuality. Their character does not change. That is reason why that recessive character again repeats the F2 generation, even though in F1 generation it is disappear. Recessive character is disappear in F1 generation, but it is appears in F2 generation because that recessive gene is retains its individuality. It is not altered by the dominant gene, even though it is present along with that. The next concept is incomplete dominance. So definition of incomplete dominance. When two different alleles are together, neither one of the alleles is fails to become complete dominance and they produce intermediate character. This is known as incomplete dominance. We are all very well known that dominance, that means one gene suppress the another gene and it express its character, that is called as dominance. But incomplete dominance means the one of that alleles, that means two genes are present, in that one of the gene is dominant over another gene, then that character is expressed. If it fails to dominate on the other allele, it can give the intermediate character, then it is called as incomplete dominance. Incomplete dominance is also called mosaic inheritance or intermediate inheritance or blending inheritance or partial inheritance. These are all the other names of that incomplete dominance. This incomplete dominance was discovered by Correns in 1903. It is not an example of pre-Mendelian concept. Remember, pre-Mendelian, that means it is not example. Before Mendel, it is not discovered. It is a post-Mendelian concept only. Pre-Mendelian means before Mendelian period. But this is a concept. Example for the concept of incomplete dominance is after Mendel concepts. So that's why it is not an example of pre-Mendelian concept, because the parental types reappear in F2 generation. It is considered to be an example of quantitative inheritance where only a single gene pair is involved. So let's know that definition of this quantitative inheritance. Quantitative inheritance. An inheritance of a character that depends upon the cumulative action of many genes, each of which produces only a small effect. Incomplete dominance is shown by the flowers of snapdragon. The scientific name is Antherinum manjus and it is commonly called as dog flower. And another flower which shows that incomplete dominance is 4 o'clock plant, that is Mirabilis jalapa. That is scientific name of that 4 o'clock plant. There are two types of flower color in pure state, that is red and white. In that plants there are two types of flower colors we can observe in pure state, that is red and white. The plants with the red flowers, capital R, are dominant over plants with white flowers, small r. One is dominant, red is dominant and white is recessive. When two types of plants are crossed, the hybrids or F1 generation have pink flowers. So let's come to the explanation of that. So here it is, a female parent that we can consider as a capital R red. It is a dominant character. It is dominant over. It is dominant capital R, and it is cross pollinated. It is a cross pollinated with a recessive character, that is white. So this white is the recessive character. Recessive character. When these are crossed, that means the gamete production, that is a gamete with capital R and another one gamete with a small r, and they are restored into pair condition, that is during fertilization, during gamete formation. Gamete is formed after fertilization and it gives the F1 generation. So up to here it is application of that monohybrid cross. Instead of tall and dwarf we are using the red and white. So red is dominant, that's why capital letter is, that means capital R. So already we know that recessive we need to mention with the small letters of that, small r, small r. So F1 generation, it is capital R, small r. As per monohybrid concept, capital R and small r should need to become, need to express red as per one gene concept proposed by Mendel. Is it correct? Correct only, no? So, as per monohybrid cross, the capital R, small r represent the red because capital R is a dominant, that is red color. So that should be expressed. But what we are discussing here, incomplete dominance, it fails to dominate over this recessive character. That's why it gives the intermediate color, not red color, that is pink. All F1 generation individuals are pink color. So this heterozygous capital or small r represent the a new character, that is pink. It is due to the incomplete dominance. I think you may understand. When plants of F1 generation, that is hybrids, or self-pollinated, then they produce the two different types of gametes because of its heterozygosity. So when plants of F1 generation are self-pollinated, they produce two different types of gametes because of its heterozygosity. So we are all well known that when two F1 individuals or hybrids are self-pollinated, we will get F2 generation. So the plants of F2 generation are of three types. So let's see that Punnett square, that F2 generation. So capital R, small r of one male parent and female parent, capital R, small r. So when these are fused, that is capital R always indicates the red and small r always indicate the white. Already we discussed, due to failure of dominance over recessive, it gives the intermediate color, that is pink, that is capital R, small r, capital R, small r. These two are represent the pink color. So that means we are observing three types of plants. The plants with red flower is a one, and plants with the pink color two, and plants with the white color flowers are three types of plants. That means three types of flowers in that plants we can observe. The plants with three types of flowers we are observed. Let's come to the conclusion of this incomplete dominance. The proportion of the plants with red flowers 1, plants with pink flowers 2, plants with white flowers 1. Therefore, so we are saying that plants with red flower, red color by visibility phenotype, plants with pink flowers phenotype, that means externally, and plants with white flower, again it is. And if you know already the genotypes also, red flowers are dominant, that is always capital R and capital R. Pink flowers, that is heterozygous condition, capital R and small r. White, small r, small r. That means the genotype is also 1, 2 and 1, and phenotypes are also 1, 2, 1. So therefore the phenotypic ratio in incomplete dominance is 1 is to 2 is to 1. So that means one plant with the red color flowers and two plants with pink color plants and one plant with white color flowers. And phenotypes. So one plant, that means with the red flowers, are always with capital R, and two plants with the pink flower are having capital R, small r, and one plant with white flowers having small r, small r. So this is a speciality in incomplete dominance. So in incomplete dominance, both phenotypic and genotypic ratios are same, 1 is to 2 is to 1. So once recollect that monohybrid cross by using tall and dwarf, there the phenotypic ratio is 3 is to 1. That means three plants are tall and one plant is dwarf. And genotypic ratio is 1 is to 2 is to 1 genotypic ratio. That means this one plant having capital T and two plants are having capital T small t and one plant having small t small t. So that means 1 is to 2 is to 1. That is genotypic ratio. But in incomplete dominance, when they are present in heterozygous condition, the dominant gene failed to dominant over the recessive gene. That's why it gives the intermediate color, that is pink color. So that's why the phenotype and genotypes of this incomplete dominance are same, 1 is to 2 is to 1. The next concept, that is codominance. Definition of this codominance: when the alleles for two contrasting characters are together in heterozygous condition, both the alleles express their character together is called codominance. So remember, if both genes are expressed their character, both. Till now we discussed only one in heterozygous condition, capital T small t or capital R small r. In that heterozygous condition, one capital T small t, in that only capital T is expressed in character. And in second, capital R and small r, it fails to dominate over small r recessive. That's why intermediate color is appeared. But in this codominance, in heterozygous condition, dissimilar factors, in the dissimilar factors, both the factors will express their character. Then it is called as codominance. Example: AB blood group in man, and another one is coat color in rabbit. Coat color in rabbit. Now, ABO blood groups are controlled by the gene called I gene. ABO blood group controlled by a gene I gene. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface. Protrude means that will slightly comes out from the surface, and the kind of sugar is controlled by the gene. That sugar polymer is controlled by that gene I. The gene I has three alleles. Three types of alleles are present in this gene I, that is IA, IB and IA. Sorry, and small I. So three alleles are present. One is IA and another one is IB and another one is small I, or the representation of the three alleles, representation of this gene. So here the I is represented. What it denotes? I denotes that isoagglutinogens. The I denotes the letter I represent. We are saying that a sugar polymers, that is iso. The I represent isoagglutinogen. So this I indicates this compound name isoagglutinogen. So this I gene represent with the three alleles IA, IB and small I. In some books, this AB blood group, instead of I, they are using the letter L. The gene is represented with L instead of I in some books because of honor towards the Landsteiner. He is the scientist discovered the blood group Landsteiner. He discovered the blood groups. So that's why he is the father of blood groups, Landsteiner. To honor him, in some books, this gene is represented with the L or maybe I. So if it is represented with I, that indicates isoagglutinogen. If it is represented with L, that is the scientist name who discovered that blood groups, Landsteiner. So we are already discussed that I gene, it is a control over that sugar polymers. So now here the alleles, I gene having three types of alleles, IA, IB and small I. So now IA and IB produce the sugar polymers. They are slightly differ, but both are produce the sugar polymers. But the third allele, that is small I, does not produce any sugar. So now here humans are diploid organisms. That's why the person possesses any two of the three alleles. We are already studied that the factors, that is the alleles are present in pairs. So that's why the cell consists, we are mentioned total three, IA, IB and small I. Among these three, only any two are present in the cell. Any two. That means the cell may consist IA and small I, or IB small I, or IA IB, or IB IB, or IA IA, or small I. So any one of this combination. So here IA small I, this is one pair. IB small I, this is another pair. IA IB, this is another pair. IB IB, this is another pair. IA IA, it is another pair. And small I and small I, this is another pair. So any two of these three allele present in a cell. Now when IA and small I are present, IA expresses because I does not produce any sugar. So that means when IA and small I are present in a cell, the small I is recessive because it do not produce any sugar, and IA produces that sugar polymer. That's why IA is the dominant and it will express its character. And similarly, when cell consists IB and small I, again IB expresses because the IB is also produces the sugar. polymer. That's why it is dominant. So IA and IB both are dominant over small I. If both combination IA and small IB and small I, in that combination, always that respective IA or maybe IB expressed. I does not express anything because it does not produce any sugar. Now come to the conclusion of that. When IA and IB are present, IA, IB are present together, both are having ability to produce sugar polymer. That's why this IA and IB both are expressed their character, that is AB blood group. A, B blood group we are saying. So we have blood groups A is there, B is there, AB and O blood group, four blood groups. In that AB blood group is the codominance because both the genes are expressive their character. So that's why AB blood group is considered as codominance. Thank you, dear students. The principles of inheritance and variation comes under concept genetics. So the genetics are always puzzles if you are not understand properly. So that's why please listen carefully and make a proper notes and work on it. Otherwise, they are always puzzles. Thank you.